our lecture is integumentary system. It is represented by the skin and its appendages. So we start directly from the structure of the skin. It is represented by two layers, the epidermis and dermis. They derive from different embryonic germ layers. They have different origin. So epidermis originates from the ectoderm, while dermis from the mesoderm. And epidermis is represented by the epithelial tissue, which is stratified squamous keratinized epithelium, and we'll investigate all layers in detail a little bit later. And dermis is represented by connective tissue, but there are two layers: papillary, which is close to the epidermis, and they interpenetrate each other. So papillary layer is represented by the loose connective tissue, while the reticular layer is represented by the dense irregular connective tissue. And to understand what's the difference between them, you have to remember the first semester topic, loose connective tissue. And from the classification, you remember that dense connective tissue, it contains more collagen fibers comparing with the loose, and regular and irregular differ by the orientation of their fibers. So if the tissue is irregular, means that collagen is oriented in different directions, not parallel. And in our dermis, this organization helps to prevent tearing of the epidermis in different directions. We can stretch it, but it is very difficult to tear, because collagen prevents that. You remember that collagen, it has two main properties. The first is the mechanical strength, tensile strength. It is very difficult to tear. And the second is water retention. So our skin is moisturized, not because water is absorbed from outside, but instead because here collagen absorbs water and retains it associated around its fibers. And also, except collagen, there are lots of ground substance components like hyaluronic acid together with proteoglycan aggregates. They contain sulfated glycosaminoglycans, which also polar molecules, and they retain water. So they're widely used in cosmetology. The synthesis of those substances are artificially stimulated by different molecules, as well as they are introduced, like hyaluronic acid is directly introduced into the dermis to prevent wrinkles formation. So this is not the topic of our lesson, because this is the topic loose connective tissue, and if you want, return to the previous semester lectures. Now let's move on and consider in details all those layers of the epidermis, and after that different structures associated with the skin, like hair, its associated glands, many other different things. Okay, so epidermis is a very thick layer and very thick epithelial tissue. It is devoid of blood vessels, as you remember from the first semester. So there is the gradient of nutrients. The basal layer is exposed to blood vessels present in the papillary layer of our dermis. And moving upwards, cell, they lose nutrients and eventually they die in the superficial layers. Among those cells, we can distinguish keratinocytes, which are epithelial cells, and different other subtypes like like Merkel cells, melanocytes, and Langerhans cells. And a little bit later, we'll consider them all. And let's start to investigate epidermis layer by layer, starting from the base. So at the base of the epidermis, there is a so-called stratum basale, the basal layer. It is also called stratum germinativum, as it gives rise to all the other layers. Here exactly stem cells are located, and by division they move upwards, replacing the cells of the uppermost layer. Those cells are cuboidal to columnar in shape, and it is important because we say that the epithelium itself is squamous. But cells in this epithelium, they are different in shape. They differ in shape, and we call the epithelium squamous only because of the superficial layer. Only because of this layer we classify it as a squamous. Okay, let's move on. And the next layer is called stratum spinosum. As cells present here, they exhibit so-called spikes on their surface. and those spikes are formed due to desmosomes, which are present between the cells. And it is important to remember the topic of the first semester, intercellular junctions. There are several subtypes of them, and desmosomes, exactly, they are responsible for the mechanical strength. Again, epidermis is very difficult to tear, as well as dermis, and that's exactly due to mechanical coupling of cells via their cytoskeletons. Let's look how those junctions are formed. In this fluorescent micrograph, that cytoskeleton of cell is represented. So here keratin filaments, which are intermediate filaments, and they are the part of the cytoskeleton. You remember there are microtubules, microfilaments, and intermediate filaments. So keratin is exactly the intermediate filament, and you see that it is present all over the cell, and also it sends small processes from one cell to another, and they are something like shaking their hands, and those hands are the projections of cytoskeleton made up of keratin filaments. And you see those red or yellowish dots. These are desmosomes, like pricks. They are used to contact and to just fix one cell to another. So to disintegrate this contact, we have to disintegrate two cytoskeletons. It's very difficult to do. So desmosomes are like contacts between the two skeletons of the cell. That's why it is so firm and stiff. And here the transmission electron micrograph is given, showing us really that between the cellular protrusions and processes there are desmosomes. They're characterized by the increased electron density, because here aggregation of keratin filaments seen. So in the stratum spinosum, cells they form so-called spikes made up of cytokeratin filaments, and those spikes are formed due to desmosomes because they are the part of the intracellular junctions. And it is better to understand the function when we see its violation. So in this case, in case of autoimmune diseases, antibodies are formed against desmosomes. So desmosomes are broken, and as you can see, the consequences, the skin is covered with the ulcers because epidermis is sloughed from the surface, because cells can't hold one with another because desmosomes are disintegrated. So you can see that the normal function of cell desmosomes is to hold them with each other. And it's very important because skin is always exposed to mechanical stress, especially in our palms and soils. Okay, let's move on. And if we are talking about the cells of the first two layers, we should remember the function of the skin is vitamin D synthesis. Exactly keratinocytes of those two layers, under the action of the ultraviolet radiation, they produce the previtamin D3. After that, it is moved to the liver, where it is hydroxylated, you remember, in the cells of the Kupffer cells. And after that, it is moved to the kidney, where it is hydroxylated one more time, and after that it becomes eventually active. So when we talk about kidney, next topic is urinary system, we'll again return to the vitamin D3. This vitamin acts on the skin itself, as keratinocytes, they have receptors to this vitamin, and deficiency of the vitamin or its receptor defects results in problems with the hair growth and different problems with the skin. Okay, so let's move on. Next layer is the stratum granulosum. So here cells, they start the process of keratinization by producing two types of granules. From its name, we guess that cells are filled with granules. And if we look at the specimens, this specimen besides them, we'll find out that really stratum granulosum, it differs from all the previous layers, as it contains densely packed basophilic granules. So what's there in those granules? There are two types of them. First is keratohyalin granule. first type, it is produced in free ribosomes, so they are not surrounded by the plasma membrane. And those granules, they contain two types of proteins, filaggrin and trichohyalin. And it is not really difficult to remember if you understand the essence. Yeah, filament aggregation. This protein helps to aggregate keratin filaments to make them more thick, while trichohyalin, also participating in the same process, it consists of two words. Trichos means hair, and hyalin like glass-like hyalin structure, homogeneous structure. So, keratohyalin granules, they help to aggregate cytoskeleton into thick bundles called tonofibrils, and in this way they make our superficial cells dense and scale-like. And the second part of granules, these are lamellar bodies. They are surrounded by the membrane, and they're destined to be exocytosed eventually, and they contain different substances, so-called probarrier lipids, processing enzymes, and proteases. The function of proteases we'll discuss a little bit later in the stratum corneum, but now we have to understand that our skin is impermeable for water, and it is especially important as we do not live in the water like fishes, and in evolutionary development of different organisms and species, this barrier formed by stratum granulosum, which is called water barrier of the skin, is very important in preventing evaporation of the water on the one hand, and on the other hand it prevents entrance of water-soluble substances inside the organism through the skin. So skin is impermeable for water-soluble substances, and if we are swimming in the water, it doesn't mean that water penetrates our skin and enters its inner environment of our organism. So let's investigate this barrier in details. Besides here, lamellar bodies are shown, and you remember previous lecture we had respiratory system. There were also lamellar bodies in type 2 pneumocytes, yeah? And they contain the surfactant components. Here, lamellar bodies are called so also because here phospholipids are aggregated in membrane-like plates. So many layers of phospholipids are present there, but their function also to be exocytosed, but they produce not surfactant, but the component of the water barrier. So water barrier of our skin is represented by two components. The first is produced by keratohyalin granules, and it is represented by dense proteins deposited under the cell membrane. So in every cell of our epidermis, in terminal stages of keratinization, dense proteins are deposited under the membrane, forming this dense shell, and it helps epidermis to perform its mechanical function, its mechanical protection. The second component of the water barrier is the lipids surrounding the cell. So lamellar bodies, they are exocytosed, and they cover and surround every keratinocyte from outside. And those lipids, they fill spaces, they cement the extracellular space. So between the keratinocytes, there is no space because it is filled with a lipid-like substance produced by lamellar bodies. So inside the cell and outside the cell, there are two different groups of substances helping to prevent water from crossing the epidermis. So once again, keratohyalin granules, they contain different substances, but just keratohyalin. It contains trichohyalin and filaggrin, and they help to make those keratin filaments dense. They transform them into dense tonofibrils, and those tonofibrils they occupy all the inner environment of the cell, making it dense. While lamellar bodies they produce a substance surrounding the cell, and those two elements are very important important in providing this barrier. Okay, with the red color, the major components of those two barrier are labeled. Okay, so the next layer of the skin is the stratum lucidum, just after the stratum granulosum. It is present only in the thick skin of our palms and zoils, because here epidermis is very thick, and we can discriminate this red oxyphilic line between the stratum granulosum and the stratum corneum. But in the thin skin, in other places of our body, it is indistinguishable, and it contains some dead cells. Cells are already dead, organelles are destroyed, expelled, and it has its like shiny appearance. Then here, besides, the difference between the thin and thick skin is shown. Notice that when we say thick skin, we imply thick epidermis, and this is not correct, but it was used to say thick skin and imply thick epidermis, and you just have to remember this. So if we are talking about the thick skin, we do not imply the thickness of the dermis. We do not take into account. So there is the paradox, yeah, because thick skin might include thin epidermis, and we call it thin skin. Okay, just remember that. Okay, so in the thin skin, in the thin epidermis, stratum lucidum is absent, while in the thick skin, mean thick epidermis, when the epidermis is very thick, stratum lucidum can be distinguished. Then, so the uppermost stratum corneum, what happens in this layer? Cells are already dead, as in stratum lucidum, so organelles are indistinguishable, but what's the difference? Unlike stratum lucidum, in the stratum corneum, cells, they start to slough off the surface, so we are losing them continuously, and that's due to breaking down of desmosomes. And before discussing that, we have to say that keratinization is a process of keratin filaments conversion into tonofibrils. We have already discussed them. So here cells are dense, they are stiff, firm, and as they are so stiff, they participate in the water barrier. So those thick proteins, they make cells impermeable for water, so water can't pass across the cell. Yeah. And what is especially important here, cells are exfoliated or sloughed from the surface because of the action of special enzymes, which are called kallikrein-related serine peptidases. Those enzymes are produced in the stratum granulosum, but they are inactive because notice that there is the pH gradient between the stratum granulosum and the stratum corneum. Moving upwards, we see that pH decreases. So here a little bit acidic environment is present. And as it is shown, kallikrein-related serine peptidases, KalK, here they are those peptidases. They are blocked with a special substance, but under the low pH they dissociate one from another and they become unblocked. So they start to break down desmosomes. Why it is important to understand? Because now you understand if the pH gradient of the skin is distorted due to some reasons, then those peptidases might become active earlier and destroy the intercellular junctions between alive cells and causing different problems. Particularly the skin is more sensitive for different hypersensitivity reactions, and atopic dermatitis might occur, for example. Okay, so remember, those peptidases breaking down desmosomes are produced by the cells in the stratum granulosum. They are exocytosed together with lamellar bodies, and here they reside in inactive state until they reach the superficial layers where pH is lower, and here they dissociate from their inhibitors and start to break down desmosomes. As you may see, even just notice that even when cells are already dead, even when all organelles are destroyed, desmosomes are still holding those cells one with another. So firm those junctions are, that even after the death they provide this mechanical coupling. Okay, let's move on. And after we have considered all layers of our epidermis, the process of keratinization and the water barrier, we have to discuss another cell populations, as except keratinocytes, there are some other cells. Of course, keratinocytes, they represent nearly 90% of all the cells, but up to 5% are represented by melanocytes. And these are very interesting population, as imagine they derive from the neural crest. So in embryonic development, you remember, when neural tube neurulation takes place, those ridges, they fuse one with another, and by each side of the neural tube, the groups of neural crest cells are present. So those cells, they migrate and populate our skin. And here they reside without making desmosomes with neighboring keratinocytes. They form lots of protrusions. That's why they are sometimes called dendritic cells. And they produce melanin. Melanin, which is the dark brown pigment protecting our skin from ultraviolet radiation. And first of all, we have to say that melanocytes, they are associated with a given number of keratinocytes. And this ratio is more or less stable, and it doesn't depend on the nationality, on the race, or on any other parameter. So in the dark and light skin, independent, there is the stable ratio between melanocyte and keratinocytes. So their complex of one melanocyte and associated keratinocytes is called epidermal melanin unit. And the question is, what is then the difference between the dark and light skin? The difference is not in the amount of melanocytes, just fix it, and it's very important. The number of melanocytes is equal, but melanosomes produced by melanocytes, the amount of pigment differs. Of course, pigment is more abundant in the dark skin and less abundant in the light. And as melanocytes, they reside in the stratum basale, they supply with melanin different types of keratinocytes. Moving upwards, those melanin granules become disintegrated, as well as all the other cellular organelles. So in the stratum corneum, where cells are already dead, they don't have any other organelles, and melanin is also destroyed. But in the dark skin, melanosomes, they are protected from degradation. So here we can see melanin in all layers, while in the light skin, melanin is present only in the first two layers, and it is destroyed in the uppermost. Why it is important to take into account? Because light and dark skin, they have the advantages and disadvantages. Light skin, for example, it has less melanin, and if such people are living near the equatorial plane, in those latitudes, the ultraviolet radiation is more active, so such skin develops skin cancer more actively comparing with the dark skin, which is better protected from the ultraviolet. Yeah? But if we are moving to the northern latitudes, then dark skin, it contains too much melanin, and it protects from ultraviolet to such a degree that keratinocytes of basal layer, they can't produce enough amount of vitamin D3. So they suffer from deficiency of vitamin D3, and it should be taken into account, especially for children with a dark skin living in the north. Yeah. While light skin, it contains only a small amount of melanin, it produces vitamin D3 more actively. So there are some advantages depending on the region and disadvantages. Okay. So let's move on and discuss some details about the melanocytes. First, they produce melanin from a special amino acid tyrosine with the help of enzyme tyrosinase. Tyrosinase is important as it is used as a marker for melanocyte and the cells derived from them. Then, how melanin is produced? Tyrosinase is produced in the rough endoplasmic reticulum. Then it is included in a special vesicles, membrane-bound vesicles, and tyrosinase start to convert tyrosine into melanin inside those vesicles. And as melanin appears inside vesicles, they are called melanosomes. So melanosomes are surrounded by the membrane and contain this pigment. But melanocytes, they produce melanosomes not for their own purposes, but they donate those melanosomes to all cells around them. First of all, to keratinocytes. And as it is shown here in this diagram, through their protrusions, they inject melanosomes inside keratinocytes, where they occupy the supranuclear position like a cap. It protects, like an umbrella, it protects nuclei of keratinocytes from the ultraviolet radiation. Why it is important, especially in the basal layer? Because here cells undergo cell division, and along with the mitosis, synthetic period takes place, and a synthetic period in the interface when implies the DNA uncoiling. And along with the DNA synthesis, DNA molecules, they become especially fragile and sensitive for any kind of damage. So those cells of the basal layer, they should be protected better than others. First of all, because they are very important, they are stem cells and they are responsible for the regeneration all over the life. And second, because they undergo mitosis, and along this process they are especially sensitive. Okay, so melanosomes are produced by melanocytes but injected into keratinocytes. Then remember that synthesis of melanin is affected by melanocyte stimulating hormone. So melanocyte stimulating hormone is produced by pituitary and as well by keratinocytes. And remember that in the pituitary we investigated such cells as basophils, and among basophils there were corticotropes, and they produced corticotropic hormone in form of a lone precursor which is called pro-melanocortin. And this pro-melanocortin, besides that's what's written here, melanostimulating hormone is produced by pituitary and keratinocytes along with pro-melanocortin and adrenocorticotropic hormone. Remember that this pro-melanocortin is the large precursor for both corticotropic hormone and melanocyte stimulating hormone. So this pro-melanocortin, when it is cut into pieces, it gives rise to corticotropic and melanocyte stimulating. So they are connected one with another. And in endocrinology, further, you'll investigate different syndromes when the problems with corticotropic hormone are accompanied by hyperpigmentation of the skin, not due to the suntan or ultraviolet exposure, but due to the increased amount of melanocyte stimulating hormone. So remember this association, that this hormone is tightly connected with the corticotropic hormone, and disorders of one is accompanied by the disorders in another. Okay, so this hormone acts through the melanocortin receptors on the melanocytes and stimulate them to activate tyrosinase to produce more melanin. This is the mechanism of the suntan. When ultraviolet activates keratinocytes, they stimulate those melanocytes through the melanocyte stimulating hormone. And this hormone also stimulates the DNA repair. And in this way, it protects cells from ultraviolet radiation by producing melanin and also by stimulating DNA repair. So there is some direction in medicine when the suntan is caused not by the exposure to ultraviolet, but melanocyte stimulating hormone may stimulate the production of melanin and protection of the skin from ultraviolet in this way without exposing to the ultraviolet. And it is used for fair skin. Okay, never mind, let's move on. So here the specimen is shown where cells in the stratum basale contain melanin. Not all of those cells are melanocytes, but they are melanin containing cells. So remember that melanin might be revealed in different cells, not only in melanocytes. Okay, problems with melanin synthesis. There are lots of them, like albinism, where it is not produced. Then vitiligo. This, due to unknown purposes, melanocytes are absent in some regions of the skin, for example, due to autoimmune projection. And you know, with aging, the hair grain also is due to okay, we'll talk about it a little bit later. So after we have discussed those melanocytes, we go to the next subtype and population of the skin. These are the Langerhans cells. They are antigen-presenting cells, and the first question to be considered is the origin of those cells. In many textbooks, it is still written that they derive from monocytes circulating in the bloodstream, but actually that's not true, as it was revealed more than 5 years ago that really such cells as microglia in our brain, as well as Langerhans cells, they populate our epidermis in embryonic development. They arrive from the yolk sac and from the fetal liver. As you remember, in the fetal period, liver is used as hematopoietic organ. So, and they arrive to the skin, reside here, and stay for the whole life. So monocytes, they can, what is written here, under steady state, Langerhans cell, they can maintain their population by division, but after acute injury, their pool might be replenished by monocytes of the bloodstream and by bone marrow derived cells. So really, bone marrow derived cells, they might form some percentage of Langerhans cells, but the majority of Langerhans cells, they are present in the epidermis before the birth. It is important to take into account. Okay, so what function they perform? They expose major histocompatibility type 2 receptors, and you remember these are receptors for antigen presentation, and they are exposed on antigen presenting cells, so they can launch the adaptive immunity. Then CD1A receptors, they are for lipid-borne antigens, and a special lectin called Langerhans. We'll talk about it later. And what else? Their main function, they phagocytose engulf the antigens possibly present in the skin if they try to penetrate the skin. And after that, they migrate to the regional lymph nodes, presenting those antigens to T lymphocytes and trying to find those with the cognate receptors on their surface. So Langerhans cells, they used not only like scavengers, but also as cells stimulating and launching the adaptive immunity. Then Langerhans cells, they might give rise to tumors, as well as keratinocytes, melanocytes. Besides melanoma, I forgot to say, this is one of the most malignant tumors, as melanocytes, they migrate for long distances in the embryonic period, so they readily give metastasis, as they are not fixed in the skin by desmosomes. Langerhans cells, they also can divide, increase in the number, and this is what we call histiocytosis. So you also should keep in mind that every cell population could give rise to different type of tumors. So a little bit more about Langerhans cells. Under electron microscope, we can reveal a special characteristic feature. These are Birbeck granules, tennis rocket-like vesicles. The function is not very well understood, but it is assumed that they are the results of endocytosis. And as Langerhans protein is present inside, it is thought that Langerhans, it helps to entrap antigens, engulf them, and after that it participates in their processing. So just remember that. Okay, about Langerhans, except Langerhans cell, in our epidermis there are a lot of other cells. In the epidermis there are CD8 killers. In the dermis there are lots of CD4 helpers, and this is not the subject of our lecture, but I'll put the hyperlink in the description below for a very helpful video about the skin immunology, as well as for melanocyte, their action. Okay, so next type of cells, this is the Merkel cell. These are the cells with unknown derivation, as it is written in your textbook, but I don't understand why the derivation is unknown, because in the 2009 it was published an article with more or less trustful results, evidencing that Merkel cells, they derive from the same source as epidermis, so from the ectoderm, not from the neural crest. So I think the derivation is known, and they derive from the epidermis. So those cells, they are used as mechanoreceptors, as they have the synaptic contact at their base, and what is written here? The combination of the synapse and the Merkel cell is called Merkel's corpuscle, and it is the sensitive mechanoreceptor. Yeah, the light touches are perceived by this cell. Then also they contain granules with the neurosecretory peptides, and they are listed here. We do not ask you to remember all of them, but just remember that those granules are present, and it is believed that granules are used as neurotransmitters as well as for paracrine action as hormones. So Merkel cells are thought to perform double function as endocrine cells as well as for nerve signaling, yeah, as mechanoreceptors and hormone-producing cells. Merkel cells, they also might give rise to Merkel cell carcinoma, which is a rare tumor but malignant. And just summarizing the information about the skin, about the epidermis, we have to remember all those receptors present in our skin. We considered them in the topic nerve tissue, but now let's refresh our knowledge. So Merkel cells, they are responsible for the tactile sensitivity. Also, there are some free nerve endings in the epidermis. They're responsible for the pain. Then different receptors perceiving the touch. Light touch is perceived by Meissner's corpuscles. They are located in those epidermal papilla of our skin, while pressure and vibration is perceived by the Pacinian corpuscles, which are hidden deeper in the dermis and surrounded by a very thick capsule. The capsule is thin in Meissner, so the light touch can be perceived, while Pacinian corpuscle is very well encapsulated like an onion, in a cross section. Also we have the Ruffinus corpuscle, Krause's and bulb for the striation and perception of striation and the cold, respectively. Notice that those four types are encapsulated receptors, okay, this is the subject of physiology, I think. Then, after we have discussed the epidermis, its layers, its cells, and now we have to discuss skin appendages, and we start from the sebaceous glands. Sebaceous glands, by its classification, they are simple, alveolar, and branched. Simple means the duct is unbranched. Alveolar, branched means the shape of the secretory portion. They have holocrine secretion, and you remember that this secretion is when cells along the secretory cycle, they store the different secretory products, and eventually they die, and by disintegration they form those sebum, their secret. So the secret of our sebaceous glands is represented by dead cells. Okay, and those glands, they are associated with the hair follicle. The secretion of those glands is very helpful in preventing evaporation, in moisturizing the surface of the skin, in making it more elastic. It prevents the dryness of the superficial layer, and also prevents cracking of the epidermis. Okay, and remember the term the pilosebaceous unit. This is the unit of the skin, including the hair follicle, erector pili muscle, and the sebaceous gland, which is associated with the hair follicle. Okay, and now two types of glands, acrine and epicrine sweat glands should be considered. They produce quite another secret. They are tubular, simple, sometimes might be branched. They are coiled, and despite they both are sweat glands, they perform very, very different function, and let's consider them by comparison. First, what's very interesting is that name is given to confuse students, I think, because despite they're called acrine, epicrine, they both have merocrine type of secretion. Just remember that. Then both they have the same ducts lined with the striated cuboidal epithelium, and in case of eccrine glands, they resorb sodium. Okay, then, now as for the differences. The eccrine glands, they are distributed all over the surface, almost everywhere of the skin, and their main function is thermoregulation. So they secrete water and some amount of salts, and in this way, when water is evaporated from the surface, it provides cooling, and in this way, by sweat production, we decrease the temperature of the body. Okay, during the active physical exercises, for example. Then they are innervated by cholinergic nerve endings, and they contain light and dark cells. Light cells, they produce water predominantly, while dark cells, they contain rough endoplasmic reticulum, so they produce some amount of proteins. What is important is that in their ducts, sodium is resorbed, as I've just said. So the sweat is hypertonic. It contains less sodium in the blood plasma, and it is produced on the skin surface. If we take all those eccrine glands together and group them together, we'll obtain a third kidney. So eccrine glands, they also participate in the excretion, excreting some products out of the organism. Together with our lungs, together with our kidneys, they participate in the excretion of different waste products. But main function is thermoregulation. Okay, this is a well-known fact, and they are not associated with the hair follicle. As you can see, they open independently on the skin surface. Now, as for the epicrine glands, they look similar, but they differ. First of all, they are associated with the hair follicle, and they are formed out of the same invagination of the epidermis. Then they produce not water, but predominantly proteins and lipids, and those secretions are produced to the hair infundibulum of the hair follicle and covers the skin as well as the hair from outside. It is believed that those substances, they perform the function of pheromones, so they are very important in communication between the different sexes, in maternal instinct and so on. But of course the function is much more prominent in animals, so they use their smell more actively than humans, but in humans it also works, and there were many experiments evidencing that really the secret of those glands affects the behavior of opposite sexes. Okay, we'll talk about it a little bit later, but as if to complete, we have to complete the comparison, we have to say that they are activated at puberty by sex hormones. So if eccrine sweat glands, they work since the birth, then epicrine glands, they are activated only at puberty. Yeah, and they are under adrenergic innervation, so they are activated under the stress conditions. And they are associated with the hair follicles, and they are located in specific regions, so they are not distributed uniformly. They are in the axillary region, in the groin, in the forehead, in some special regions of our body present. So the next slide is about the vomeronasal organ. Last time we had topic respiratory system, and we remember that there is the olfactory epithelium in the nasal cavity, but also there is one more epithelium with a special function, which is much more developed here in animals than in humans, but in any way it is present, and exactly here those substances produced by epicrine sweat glands are perceived, and together those pheromones in vomeronasal organ, they are responsible for the sexual behavior, for the maternal instinct, and as for animals, they select the partner for reproduction, and they exclude some partners, potential relatives, or ill, genetically defective animals, based on the smell. They analyze the smell and they can make lots of conclusions because of that. Okay, so epicrine sweat glands, they represent quite another type of glands, and modification of epicrine sweat glands is mammary gland. So, okay, mammary gland we'll consider a little bit later. So another skin appendage, after we have discussed glands, the sebaceous and two types of sweat glands, hair follicle should be discussed. So hair is represented by the hair itself and the hair follicle, which is hidden in the dermis. And if we make a longitudinal section, we'll find here the infundibulum, then the isthmus, then the special region called the hair bulge, very important region, and the hair bulb. So the follicular bulge is the region of the stem cell location. We'll discuss it a little bit later. So if we make a cross section through the hair follicle, we will find out that the outer layer of the follicle is surrounded by the so-called external root sheet. This is the invagination of the superficial epidermis. So when epidermis invaginates, it forms so-called external root sheet with many layers. This external root sheet is separated by from the connective tissue, surrounding the hair follicle by the so-called glassy membrane. This is the basement membrane, actually, of the epidermis. Then, by invasion downwards, these epithelial cells, they also form the internal root sheet, which reaches the level of sebaceous glands. Above the sebaceous glands level, we can find the internal root sheet only here. Internal root sheet, it includes two layers. Okay, here the longitudinal section is shown once again. Another section, here we can see the external root sheet, and here is the internal root sheet is shown. So external root sheet invaginates and then returns back with the formation of the internal root sheet. It is represented by the so-called Henle's layer and the Huxley layers. So Henle and Huxley, two layers of the internal root sheet, and after that the cuticle could be seen. If we look at the hair itself, there are several subtypes: thick hair with the medulla inside, and the thin hair where medulla is absent. So in this case we consider the thick hair. So medulla is represented by the soft keratin, while the cortex is represented by the stiff hard keratin. Hard keratin differs by the number of disulfide bonds. The keratin is cysteine-rich protein, and disulfide bonds, they affect the density and the physical properties of the keratin. More disulfide bonds, more stiff keratin is, more hard it is. So in the cortex, hard keratin is present, and and also hair is surrounded by the hair cuticle. If we are talking about the hair above the skin, it contains the cuticle, cortex, and medulla. Cuticle differs from the epidermis in that cells do not slough from the surface, but instead they continue to contact one with another. So the hair elongates if the thickness of epidermis is stable, because newly produced cells they replace those sloughing of the surface, and there is the balance and equilibrium between those processes. In the hair, the process of cells do not detach from the surface, so hair elongates. There is a cyclic period of hair growth. We call this catagen, telogen, and anagen, catagen, and the telogen. So the three periods: when hair starts to grow, when it is in a steady state, and the telogen is the period when the hair follicle stops to divide and we lose the hair eventually. So the hair loss is the normal process, and some percentage of hair is lost along the life, and newly formed hair follicles appear. And the process depends on sex hormones significantly and on different other factors, for example the hemoglobin amount and many other processes. Okay, but what we have to say is that in the hair follicle there is a special follicular bulge region, and it is located between the muscle erector pili and the sebaceous gland. In the hair bulge, we can find lots of stem cells to regenerate different components of our skin. And as it is written here, there are some stem cells for the hair follicle, stem cells for the sebaceous gland, and another stem cell for the surface epidermis. Why it is important? Because when we are talking about the skin regeneration, epidermis regeneration, we have to understand that epidermis itself regenerates from the stratum basale. So if stratum basale is not affected, or if the surface of the injury is not very significant in the square surface, so the epidermis could be replaced or regenerated by the action of basal cells surrounding the place of injury. But if the surface area of injury is too large, and if basal layer is destroyed, then regeneration might be provided by those cells hidden in the bulged region, in the follicular bulge. As you can see, it is hidden deeper in the dermis. So if epidermis is removed, then but bulged regions are preserved, so they could be used to replenish the superficial cells. So stem cells, they differ by their commitment, but they can interchange each other in case if one pool is exhausted. So under normal circumstances in the bulge region, in the follicular bulge, stem cells, they migrate upwards to replace the sebaceous stem cells, and they migrate downwards, and here in the cell matrix, by dividing, they give rise to keratinocytes, eventually forming the hair. Besides, what is especially interesting, that why this hair follicular bulge is the subject of scientific interest for regenerative medicine, because here precursors of melanocytes reside. As I told you, melanocytes, they arrive from the neural crest, and some undifferentiated melanocytes are still present in this follicular bulge. So if we take those stem cells out of there, we can more easily obtain neurons out of them, because melanocyte precursors, they derive from the neural crest, so they can be more easily transformed into neurons than other types of stem cells in our skin or adipose tissue or taken from any other sources. So skin is the most easy accessible source for neural crest stem cells, and here it is documented in 2019, neural crest stem cells are obtained from human skin, and as I remember, even from dead individuals, even if they are aged. So this is a very promising source of neurons. Okay, let's move on. So we have to discuss also the mammary gland. Actually, I had to put it after the apocrine sweat glands. In mammary gland, by classification, it is compound, as the duct system is highly branched, and it is tubular-alveolar by the shape of secretory portion. It contains lactiferous ducts and lactiferous sinuses. So ducts are lined with a simple cuboidal epithelium, while sinuses are stratified cuboidal epithelium. And what is interesting is that here cells, they have two types of secretion simultaneously. Lipids are produced in form of apocrine secretion when the apical part of the cell is destroyed, while proteins are produced by merocrine type. So two types are together present here. In detail, cyclic changes of mammary gland and its the regulation of its activity by different hormones we will discuss in the topic female reproductive system. Okay, and at last, nails. Nails are also skin appendages, and in terms of nails, you have to remember different terms. Yeah. So in the nails, we have the nail matrix, which is represented by undifferentiated cells. They migrate and form the nail root, where they divide actively, and by dividing, they give rise to the nail body, which is represented by dead keratinized cells. Besides, keratin here, as in our hair, is hard, stiff, unlike our skin. Yeah, and the fold of epidermis near the nail root, part is called the eponychium and or cuticle, and it is removed usually. Then also at the root of the nail, close to the root of the nail, we can find lunula. It has a little bit another color because cells are not fully keratinized. The process of keratinization isn't over, because when keratin is mature, it becomes transparent, like in the nail body. Also, there is the free edge of the nail and the hyponychium under the nail, preventing it from different types of mechanical injury. Okay, I think this is the last slide of our lecture. So thank you for watching. Thank you for watching till the end. And if you do really like the content we produce for you, then subscribe, please. This is the best way how to encourage us to do a little bit more helpful videos for you. And if you have any comments, we are very glad to get this feedback from you in our comments. Thank you for watching.